Greetings. Welcome to Tracking Pandemics. This is Module 1.5, an Introduction to Molecular Diagnostics. Molecular diagnostic tools are laboratory methods to determine whether or not a pathogen is present in a tested sample. Some of these tools are quantitative, some are semi-quantitative, and others are purely qualitative. We are going to focus on quantitative tools in this module. These tools can enable the determination of either whether or not a pathogen is present or how much pathogen is present, which you might call pathogen load. Molecular diagnostics are at the heart of a field called molecular epidemiology. This field is concerned with the surveillance of disease factors using molecular diagnostic tools. The primary goals of molecular epidemiology to measure geographic and temporal distribution or distribution over time of these disease risk factors. A second goal is to understand the transmission, evolution, and population structure of a particular pathogen. Molecular epidemiology, as the name implies, is analogous to more traditional epidemiology, which is also interested in the distribution of disease risk factors. Molecular epi is more concerned with the molecular characteristics of a particular infectious disease pathogen. As an example, in this figure here, this is a spatial temporal distribution or distribution over time and space of Yersinia pestis genotypes. This is the pathogen that causes bubonic plague. One of the oldest methods for determining if a pathogen is present involves laboratory culture of the organism itself. In this method, a sample that is thought or suspected to contain the pathogen is obtained, and that sample is used to inoculate the culture media or growth media for the particular pathogen of interest. This growth media is usually some kind of nutrient formulation tailored to a specific organism or more commonly class of organisms. In some cases, a generic growth media can be used, and specific phenotypic characteristics of organism can be used to determine the presence of the pathogen. Those phenotypic characteristics may be a particular type of colony formation, color, size, or growth rate. One common example shown here on the right side of the slide is called blood agar. Certain pathogens, like Corynebacterium or Klebsiella, produce toxins as they grow. Those toxins are capable of lysing blood cells. These are so-called hemolytic toxins. Blood agar is a nutrient-rich medium that also includes blood. We can distinguish hemolytic colonies by the appearance of a clear zone around them as blood cells in the vicinity are destroyed. We can then use this phenotypic response to indicate the possibility that a hemolytic pathogen is present. There are some drawbacks to this method, of course. First, growing pathogens in culture can be time-consuming and laborious. But more importantly, only a very small fraction of microbes can actually be cultured. And so we are restricted in the types of pathogens we can identify using culture techniques. Another more specific method is called the ELISA, or the enzyme-linked immunosorbent assay. In an ELISA, antigens from a particular sample are adsorbed onto some solid surface, usually a slide or a dish. They are then bound to an enzyme-linked antibody and detected with an enzyme substrate. that causes some reaction that usually induces a visible change, such as color. This is a very common assay. It has several advantages. First, it is relatively specific for your pathogen of interest. Second, it can detect signal in crude lysates or other mixtures. In other words, you don't have to purify anything. However, there are some drawbacks. First, these ELISAs tend to be prone to false positive signals where you have nonspecific binding. And second, it requires advanced knowledge of the antigen of interest and development of an antibody. A more general class of antibody-based diagnostic tools are called serological assays. You've probably heard this term kicked around quite a bit lately. In a serological assay, a blood sample is tested for the presence of antibodies that have developed against a particular antigen. So in a serological assay, we take a sample from a patient or even the environment. We incubate that with some pathogen component that's bound to solid support. We call this component an antigen. This is something that can elicit immune response. Then we add a secondary antibody with some reporter attached, fluorophore, enzyme, etc. The concept here is that if a patient or an environment has encountered a particular pathogen and an immune response has been raised, then antibodies to that pathogen will be circulating in the blood. Those antibodies in the blood of the patient will recognize the antigen from the pathogen and bind to it, and a serological assay will detect that. A few of the drawbacks: again, serological assays do tend to be prone to false positives. It again requires knowledge of the antigen. And importantly, a serological assay is generally used to test for past evidence of an infection. In other words, a patient's immune system has already detected, raised antibodies, and is now hopefully protective against a particular pathogen. Serological tests, though, are heavily used in a number of aspects of infectious disease outbreak response. First, we want to determine if a patient has ever encountered a particular pathogen. This can be done during an active illness, though not in the early stages, or on a more routine basis. For example, to determine if a patient may have been infected with the pathogen without realizing it during the infection. This application requires just a binary decision, yes or no. As such, it can be easily developed into tests such as the one on the right side of this slide, where a simple color change can determine whether there's antibody present against a pathogen antigen. These tests can even be home tests. Several of the related serological tests are being developed for COVID now. These are very scalable and easy to use by design. Serology, however, can also be used to determine the strength of a patient's response to an infection. We do that by measuring what's called the titer, roughly how many antibodies against a particular antigen are circulating in the blood. This titer can be used to infer how long a patient might be protected from reinfection with that pathogen. Circulating antibodies provide a much stronger initial response to an infection. These are the reasons why we are relatively immune to reinfection by the same pathogen. As the antibody titer wanes, usually over time, we may become susceptible to reinfection. Antibody titers can also measure different elements of the immune response by using a secondary antibody that binds to different primary immune system components, for example IgG or IgE. These quantitative assays are very powerful, but they're not nearly as scalable as the binary assays. They often require specialized lab equipment and expert interpretation. Okay, moving on to nucleic acid-based tools. Again, you've most likely had recent experience with some of these nucleic acid-based tools. Here I've listed just 6 of the tools we have available. We are going to talk more specifically about the 3 in red: multilocus sequence typing, polymerase chain reaction, and loop-mediated isothermal amplification. These are not the only nucleic acid-based tools. There's a lot of creativity in developing these tools. We'll start with multilocus sequence typing, or MLST. MLST is a useful technique to track the spread of a pathogen over a relatively short period of time. In MLSTs, a panel of conserved genes from a pathogen are sequenced and then compared to a panel of known sequences. It is based on the assumption that highly conserved genes will be present in all variants of a pathogen. In other words, that pathogen will not lose any of those genes. It also assumes that those genes will change over time and that those changes will be somewhat regular. You may have heard this referred to as a molecular clock theory. For example, the 16S ribosomal rRNA gene in bacteria fits all of these assumptions. It's highly conserved across all bacteria. It changes over time, but relatively slowly and relatively regularly. So by counting the number of changes in this conserved gene, we can get an estimate of how long it has been since it diverged from a particular neighbor or parent genotype. By comparing variability within a panel of genes instead of just a single gene, we can make better, more definitive temporal relationships between isolates of a pathogen. In other words, pathogen isolated from different patients at different times in different places. This is one of our key goals for molecular diagnostics, this definitive spatio-temporal distinction. There are several different flavors of MLST. One of the more common ones is called MLVTA, where we compare variable tandem repeats instead of conserved genes. In some pathogen genomes, there are areas of the genome comprised of tandem repeats, fairly short repeats of the same sequence, right next to each other. As pathogens replicate, the number of tandem repeats tends to slip. As such, we get different numbers of those tandem repeats in different genotypes, different strains of the pathogen. On the right, you see a diagram called a burst diagram. This example is from an analysis of an outbreak of a Staphylococcus pathogen. And here we can see the typing results, or the MLST results, clustering into several clades. Here they're labeled CC1 through CC7. If we look at CC2 in the middle of that figure in cyan, we can see that all isolates in that CC2 cluster, each of which is a circle or node, all of those isolates likely originated from the same progenitor, that gray dot above CC2. We can use information such as this to track the spread of a pathogen from person to person, or for waterborne or foodborne illnesses, determine the likely environmental source for an infection. This kind of figure here, this burst diagram, is similar to a phylogenetic tree, which measures the evolutionary relationship between organisms, but is based on more straightforward distance metrics. We can use those more straightforward metrics because we're determining spread of a pathogen over a relatively short time period. Burst diagrams are fast and scalable, so we can incorporate hundreds of thousands of nodes, hundreds of thousands of isolates in a burst diagram. The next diagnostic we're going to talk about is polymerase chain reaction, or PCR. PCR is a household term these days. It has become the cornerstone of many of our modern molecular diagnostic tools. How it works is you decide on a target sequence of interest, usually a stretch of some gene that's present in a pathogen. That target can be anywhere from about 40 to 4,000 bases long, depending on your application. Short sequences that flank this target, called primers, here colored purple, are crucial to a successful PCR. You mix your biological samples, say a nasopharyngeal swab from an individual, with the primers, a DNA polymerase, which will replicate DNA, and the raw materials needed for making new DNA, including nucleotides, shown here in green. This mix is heated up to melt or denature your double-stranded DNA in your sample, and then cooled slightly to allow the primers to anneal to your target or your sample DNA by base pairing. Those primers sit down only where their base pairs match up. Ideally, this is only outside the region of your gene of interest or target of interest. The temperature is then raised again to optimize function of the polymerase, which extends the DNA starting at those primers to generate an exact copy of your target. These temperature changes are accomplished these days in a special machine called a thermocycler. Each copy of your target can now serve as its own template and be copied itself, leading to an exponential increase in target copies from 1 to 2 to 4 to 8, 16, 32, 64, etc. By cycle n, and we usually run 35 to 45 cycles, your target has been copied 2 to the power of n times. This amplification allows the detection of targets that are in very low abundance in your original sample. The use of specific primers allows your target to be amplified from among a mixture of other things. In many cases, you don't even need to purify DNA. You just need to lyse the cells to provide access to the primers and polymerase. You can then detect the amplified product, which will dominate the DNA in your product, using some fluorescent dye that intercalates into double-stranded DNA. Alternatively, you can design a probe with a fluorescent marker attached. That probe is very similar to a primer, except its matching sequence is somewhere inside the two primers that are amplifying your product. One of the more common mods for PCR is called real-time PCR, or RT-PCR. Real-time PCR is very commonly used now in tracking infectious disease outbreaks, including SARS-CoV-2. Real-time PCR uses a special thermocycler. This thermocycler has a built-in computer and fluorescence detectors aimed at each reaction well in the machine. So as your PCR reaction progresses and product builds up, the machine detects fluorescence of your probe or dye after each PCR cycle. After some number of cycles, and that number of cycles is determined by the number of target copies in your original sample, the fluorescence in your reaction starts to increase exponentially. Here you can see an idealized real-time PCR graph. You have the cycle number plotted against, or you have fluorescence, plotted against the cycle number. As you can see, when you start out, the first few cycles, there is no detectable fluorescence. Your target is being amplified, but it's still in such low numbers that the fluorescence is not yet detectable. As your target is amplified, eventually that fluorescence becomes detectable by the fluorescence detector, and it starts to increase. In this graph, we see that start to increase around cycle 19 or 20. As this is an exponential reaction, that increase in fluorescence quickly becomes exponential. At that point, where it starts to become an exponential growth, that point is called the CQ, or the cycle quotient value. As we progress through the exponential phase, eventually the polymerase will run out of raw material to make your product, and so we will see a plateau in fluorescence. That CQ value has an inverse relationship with the number of target copies in the original sample. In other words, the more starting material you have, for example, the more copies of that pathogen gene you have, the earlier you'll be able to detect fluorescence. Instead of starting by amplifying 1 copy, maybe you're starting by amplifying 100 copies, which will then become 200, then 400, then 800. So, the more the starting material, the more copies in your original sample, the lower the CQ value. In other words, again, the more you have to start, the less time it takes to reach a detectable increase in fluorescence. So real-time PCR is fully quantitative. In other words, it can tell you how many copies of a target were in the original sample. Interpreting CQ values, however, can be somewhat of a challenge. So in this slide, we're going to walk through how we do that. The first thing we have to understand is that the efficiency of the PCR reaction plays a major role in how accurate you can quantify a particular target. We test the efficiency of a set of primers using a serial dilution of a known number of target copies. This is also called a standard curve, although it is not actually a curve, but a line. When you plot the log of the copy number, which we know in our standard, against the CQ value that's detected in your real-time PCR reactions, you can calculate a best linear fit and determine the primer efficiency. In this example, the efficiency of our primers is 99.16%. We plot log of copy number against CQ value, determine the equation of that line, y equals minus 3.3x plus 39.96. We can calculate the slope of that line, minus 3.3. And from the slope of that line, determine the efficiency of our primers. In 100% efficiency, our numbers should double. Every time a PCR reaction occurs, we should get double the number of copies. But of course, that's the ideal situation, and oftentimes we don't get an exact doubling. We can use a standard curve such as this to convert CQ values into absolute copy numbers, if we're interested in knowing that. However, we can also use CQ values as more of a positive-negative indicator. To do that, we can set a threshold and use CQ to report whether a target is present or not based on our threshold. For example, if we say a threshold of a CQ value of 35 or better indicates the presence of a pathogen, and 35 or worse indicates noise, then we have a binary test, presence or absence, yes or no. This lends itself well to high throughput applications, including time-sensitive testing, as in COVID. If we need to get reactions done quickly and answers back, we can use a binary test. However, it does not provide any additional information, such as the extent of infection, how many copies of the virus are in a particular sample. Again, many times we are not interested in this information on a large scale. In fact, many times we just want to know whether a patient has an infection, whether a patient currently harbors pathogen. Finally, it's very important to remember that CQ values are, one, inversely related to copy number. So the lower a CQ value, the more pathogen was present in the original sample. And CQ values are on a log, not a linear scale. What does that mean? That means that if the difference between 35 and 34 cycles is 10 copies, then the difference between 25 and 24 cycles, also one cycle, will be 10,000 copies. So the difference between two CQ values depends on where you are in the CQ range. If you have a CQ of 25 and I have a CQ of 24, that's an enormous difference. But if you have a CQ of 35 and I have a CQ of 34, that's a very small difference. Our final nucleic acid-based assay we're going to discuss is called LAMP, loop-mediated isothermal amplification. LAMP is a low-cost alternative to PCR. It does not require an expensive thermal cycler or other equipment. As such, it's very valuable when it can be deployed in the field, and it yields rapid results, often within 10 minutes. Those results are usually the accumulation of some colorimetric reagent. How does LAMP work? LAMP, first you generate a copy with internal primers. This is similar to PCR. But those primers contain regions of self-complementarity. In other words, one part of the primer is a complementary sequence to another part of the same primer. Those primers are used to create a product. Then we add two what are called external primers. Those primers sit outside of the internal primers. And we amplify with those external primers. When we amplify with the external primers, we are including the internal primer sequences in our amplified product. And those primer sequences have self-complementary regions. What happens is those primer regions base pair with each other, and we get a dumbbell template, what looks like a dumbbell. Two rings, one on either end of that product. The template now contains multiple sites for primer binding and initiation of replication. We quickly get accumulation of a double-stranded DNA that contains our target. It also contains extra primer sequences, but we're not too concerned about that. Unlike PCR, this can be done with what's called an isothermal polymerase. Polymerase that does not require temperature changes. One of the drawbacks to LAMP is that it's not easy to quantify accurately. So we can get crude or semi-quantitative information from LAMP, but quantitative information is somewhat more difficult. In addition, setting up a particular LAMP reaction and optimizing it can be somewhat cumbersome. Okay, moving on to the last diagnostic tool that we're going to talk about today. This diagnostic tool is protein-based. It's called the Western or Immunoblot. This is a very common laboratory method. Unfortunately, it doesn't scale well, and it takes significant time and expertise. How a Western blot works is first you lyse the cells in your sample and you separate the proteins from that sample by gel electrophoresis. Electrophoresis separates samples based on size or molecular weight. This gel is then transferred to some solid support, usually some kind of membrane, that transfers the proteins from the flimsy gel to a solid membrane. We then add a solution with primary antibody against our target protein of interest, then secondary antibody against the primary. As with ELISAs and serological assays, that secondary antibody is often conjugated to some enzyme that cleaves a colorimetric substrate, and we read out or visualize that color. So this is an assay that is specific for a protein of interest, and in infectious disease tracking, we're usually interested in a protein that's expressed on the surface of our organism. Why do we use it for tracking pandemics? Direct detection of a protein rather than nucleic acid can be crucial for some infections. A good example of this is cholera. Cholera, the disease, is caused by a protein toxin that's produced and secreted by certain species of Vibrio bacteria. In some cases, it's very valuable to know when and where that toxin is present, particularly if it might stick around after the pathogen itself is gone. In these cases, a DNA-based test may not be the best tool, but instead we may want to detect the toxin itself. In these cases, Western blot or an immunoblot may be our molecular diagnostic of choice. To finish up, we have a lot of diagnostic tools available for tracking infections. I've mentioned here just a few of the more common ones. The choice of our tool can affect the sensitivity, the accuracy, and the interpretation of the results. We need to recognize that every method, every diagnostic tool has its own particular strengths and limitations. This is particularly important to keep in mind as the results of our diagnostic tools can be used to inform public health responses or epidemiological models that can mean the difference between life and death. Thank you.